Good morning. I'm Mark Rohbal, the director of the NIH Office of Technology Transfer. It's a pleasure to have you all here today, and especially in honor of Phil Chen and his distinguished contributions to tech transfer, particularly in the early days at NIH, and especially our distinguished speakers, Cliff Berry from NIAID and Carol Nacy from Sequela. So I would, just to make a personal note, before I joined the Office of Tech Transfer 12 years ago, I worked in the Tech Transfer Office for NIAID, so I have fond memories of working with Cliff and Carol on their collaboration projects when Cliff was at Rocky Mountain and Carol was starting up locally. So it's great to see them and to honor their work. So we look forward to your story. But I want to introduce my supervisor and the Deputy Director for Intramural Research, who among his many duties oversees tech transfer, and that's Dr. Michael Gottesman. And he's celebrating his 20th anniversary as DDIR, and so we thank him for his many contributions and especially for his support of tech transfer. It's a pleasure working with him, and I thank him for all his support. So, Dr. Gottesman. One point I wanted to mention, in your brochure, there's a list of inventors. So to honor all of our inventors, people who have received patents in the last year, we've given a list by institute of all the inventors. So we want to honor them as well.
Thank you, Mark. This is a wonderful annual occasion for us, a chance to recognize Phil Chen, who was, I'll tell you in a few moments, a very important part of our intramural program for many years, and a chance also to recognize the importance that technology transfer plays in the mission of NIH, that is to do research to improve the public health. The improve the public health part requires technology transfer, and it's really important that we have stories like the one you're going to hear today in order to fulfill our mission. So this is the 8th annual Philip S. Chen Distinguished Lecture on Innovation and Technology Transfer. And the series, as I noted, honors Phil Chen, a scholar, a science administrator, a trusted advisor to many directors and deputy directors, and a friend to many of us at NIH, and many of his friends I can see are in the audience. Phil was born in Michigan, grew up in Madison, Tennessee, and in Massachusetts. He was a graduate of Clark University in physics. He got his Ph.D. in pharmacology from the University of Rochester. And at the time when he was a graduate student, he was also an Atomic Energy Commission pre-doctoral fellow, and actually spent some time with the Atomic Energy Project out in Nevada. Also as a graduate student, he co-authored a paper entitled Microdetermination of Phosphorus. And any of you in the audience who've ever measured inorganic phosphorus have used the Chen method to do that. It is one of the most highly cited papers in the history of biomedical research, certainly in the top 100. That's an amazing achievement in its own right. He did a postdoc in Copenhagen, developed a real love for Denmark and things Danish, and his wife, Inger Lisa, who is here, is a representative of that. And also here today are many of his children and grandchildren who have been faithful attendees of this series, including Kylie, who's been here ever since she was born, every year since she was born. And she's actually going directly to her postdoctoral fellowship next year. Phil came to NIH in the late 1950s, where he worked with Fred Barter, the legendary physiologist, on binding of cortisol to plasma proteins. Went back to a faculty position at Rochester, but the siren call of NIH brought him back again, where he served for 30 years, basically, in the office of the director, served 8 different NIH directors. And in most of his activities were in the Office of Intramural Research. And during my time there, he was a trusted senior advisor and a wonderful source of very, very valued information. Phil's special interests included international affairs, particularly Arctic research, and technology transfer. He, as you've heard, established the Office of Technology Transfer, and he invented our most beloved document called the CRADA, Cooperative Research and Development Agreement. And so a lot of the technology that's been transferred since wouldn't have been possible without Phil. As a senior member of the Office of Intramural Research, Phil, over the years, became known as a very caring, knowledgeable, and creative administrator. People would come to him with the most difficult and almost impossible problems, and he would find solutions. It's quite amazing. Phil, we need you now. The subject of today's lectures is TB. It takes more than a village to raise a remedy. We don't have a village here, although we have a nice community. But we do have two wonderful speakers. As you've heard, Dr. Cliff Barry, who's chief of the Tuberculosis Research Section in the Lab of Clinical Infectious Diseases at NIAID, and Carol Nacy, co-founder and chief executive officer and chairman of the board of directors of Sequela. So what I'm going to do, because they're going to do a tag team, they're so well coordinated as a result of our tech transfer process that they're going to be bouncing back and forth. So I'm going to introduce them both. Dr. Barry is an internationally recognized researcher on TB, particularly in identifying targets and developing new drugs to treat Mycobacterium tuberculosis. He has a Ph.D. in organic and bioorganic chemistry from Cornell and was a postdoc at Hopkins. 20 years ago, he joined the Rocky Mountain Lab, was out in Hamilton, as you've heard. And 15 years ago, when he was tenured at the NIH, he moved his unit called the Tuberculosis Research Section to Twin Brook near Bethesda. His group is multidisciplinary, and he has many international connections and collaborators. I think you'll probably hear about some of them. And all of that has really helped move forward the drive to develop new drugs to treat tuberculosis. Cliff, are you going to start out today? Okay, he'll make the opening comments. Our next speaker, our other speaker, is Carol Nacy. She did her undergraduate degree and Ph.D. from Catholic University, spent most of her scientific career at Walter Reed Army Institute of Research, where she studied various tropical infectious diseases, and has since moved to the private sector, where she was chief scientific officer initially at Entremed, a part-time chief scientific officer at Anergen, before she founded Sequela. She's been recognized in many ways for both her scholarly work, academic work, and for her entrepreneurship. She was elected to the American Academy of Microbiology, and she was elected president of both the American Society of Microbiology and the Society for Leukocyte Biology. Her biotech activities have been recognized in many ways as well, including being named a top 50 innovator in the U.S. and an Entrepreneur of the Year. A few years ago, she was awarded the Humanitarian Award by the Global Alliance for Immunization Against AIDS for her work to create new drugs for TB. So without further ado, I think Cliff will take the stage, and then we're going to hear a wonderful talk today. Thank you.
Okay, is this on? Yep. So I want to start by saying thank you, Michael, and thanks for the opportunity, because I don't think we really get the opportunity very often to say thank you to the tech transfer folks here at NIH, who Mark can say sort of with benefit of hindsight, he has fond memories of it, but I don't think he remembers how much work it really was. And I think I continue that trend today to really occupy a lot of time and energy from the Office of Technology Development, because of the field that I work in is inherently linked to intellectual property and to developing things. And I think it's great to have the opportunity to say thanks not only to Phil, but thanks to the entire tech transfer community here at NIH for making that possible. And I think to start out, that I want to sort of just refresh your memory of what tech transfer was intended to do in this sense and why we develop relationships like this. And so this is from the Federal Technology Transfer Act of 1986, which was an amendment of the Bayh-Dole Act that first tried to reverse the trend of the information flow from academic and government labs back into the US industry. And the quote from the hearing about that I really like is the importance of linking our unparalleled network of over 700 federal laboratories and our nation's universities with US industry continues to hold great promise for our future prosperity. So that was in 1986. Did it work? And I think the best summary of that was an editorial that appeared in The Economist in 2002 that credited that piece of legislation with reversing America's precipitous slide into industrial irrelevance, and called federal and academic labs the golden goose of innovation that sort of turns the US around competitively. And I think that's been great in some fields, and I think where we've struggled with it over the years, and what the story about this that you're going to hear about today really speaks to, is more how do we translate that into global health issues, where perhaps the incentives and the risks and the benefits of the technology that you're talking about aren't as straightforward as monetary incentives or as benefits to US industry directly. And I think one of the things to think about when you're entering into relationships like that is you really have to understand the risk and the culture in both the place that you're at, whether that's a government or an academic lab, and in the company setting or the partner with which you're entering into that endeavor. And so every different kind of organization has different perspectives on what is risky behavior and what's risk, what's the benefit of taking risk. And we, in government and academia, all sort of lump them together, think, risky stuff is inherently good science, and we should be doing risky stuff all the time. And with the caveat that in government, maybe you try to minimize that risk a little bit more. But in biotech and pharma, which are the two sectors I interact with most, they're either a little bit less, they're definitely more risk averse than academia is. And pharma is the extreme case where they're so risk averse that nothing that's essentially laboratory ready, but product not ready, is really of interest to them. And I think if you don't understand who you're partnering with through this process, then you can't translate something from the brain to a product in the end. And I think that's really what OTT's role and what OTD and NIAID's role has been, is to really start to piece together those agreements and put together partnerships between prospective industry collaborators that can translate ideas into products with groups within NIH or groups within different institutes that can actually come up with the ideas that are things that are worth turning into products. And I think they're really good at finding the right balance of things. And I can remember long conversations with Mark about the beautiful CRADA agreement. I won't say any more about CRADA as we were negotiating it, and trying to understand what went into it and all of it, the caveats and things that we tried to get right in this, so that we ultimately could take something that was an idea in the brain, right, and turn it into something that is like this. And this is really the subject of today's talk. This is a drug packaged, blister packaged for use in clinical trials that are going on now in phase 3. And I think that translation doesn't happen without setting the partnerships up right. And I think the real credit to Phil and to the Czech transfer community is that they've learned how to do that. So as I said, the subject today is going to revolve around global health. And global health really, this is from a satellite meeting that happened from the ASTHM meeting last week here in Washington, D.C., really is these days the same as America's health. There's no wall you can erect to keep people with drug-resistant TB or drug-resistant anything from flying into the US these days. It's very difficult to craft these to keep international borders with respect to infectious diseases and global health issues. And I think that the more we recognize that, the more we understand that part of NIH's mission is not just worrying about health problems that are imminent and current epidemics that happen within the US, but things that might emerge in the future, or things that we can't block from coming into our borders. And of course, I'm going to let Carol introduce the infectious diseases.
Do I have to be away from you in order
I think
to
so.
do this? Okay,
Yeah.
all right.
Yeah, at least 2 paces.
Yeah. All right, so infectious diseases are the second leading cause of death worldwide, and you are messing up my talk. How do we do this?
Intentional.
It is intentional. All right, thank you. So TB is actually in the top 10 lethal infectious diseases, and in this country, I can't tell you, I speak with venture capital firms and funders all over the country. The word tuberculosis has not been in their lexicon for 50 years, and they don't actually realize how big the problem is outside the US. But infectious diseases, they're starting to get a picture of that. And now I can at least tell them that 40% of all the deaths in the countries where we would sell product, if we were to be a successful company, are due to infectious diseases. Interestingly, we're now seeing that many of the autoimmune diseases have an infectious origin. And I think that's going to help us all in understanding more about how microbes interact with our bodies, both in a beneficial way and in a difficult way for our immune systems to understand. And I think more than anything that's scaring the public health communities around the world is the fact that multi-drug resistant organisms are coming to the fore. And we don't have enough antibiotics, and we've really paid no attention to antibiotics for a number of years. The new classes of antibiotics are few and far between. They're fairly specific for either gram-positive or mainly gram-positive organisms these days, and we're actually experiencing a crisis, both in this country and in Europe, and multi-drug resistant infections, and in particular multi-drug resistant TB is now considered a national security threat, both in the United States and in Europe. So TB is a problem on every continent. We're not without our TB. You can see that by the color of the country, how many cases per 100,000 that exist. Clearly, in areas that have the darkest numbers, we have the most number of TB patients, but we have them in the United States as well. And I don't know how many of you remember the SARS epidemic. Yes, where we were completely panicked about SARS. This came out in the New York Times in 2003, basically talking about SARS is a problem, yes, but that's SARS. In terms of numbers of people affected by SARS, number of people who died of SARS, there were at that time around 500 people who had died of SARS. At the same time, 2 million people died of TB. So the scale of our panic over certain of infections that cross our borders is unfortunate. Let's put it that way. And tuberculosis, which we have a wonderful TB elimination program in the United States, has moved TB down out of our line of sight. But outside the world, it's burgeoning everywhere. And part of the reason that it's so successful is that it's an airborne transmitted disease. And you may not know this, but the no spitting laws in New York City aren't about the fact that spitting is gross. It's about transmission of TB. So to be able to be transmitted by the air easily, you have to have a certain particle size. And that's where SARS is. And that's where TB is. So it is the perfect size to be passed person to person by sitting next to someone who is infected. And in the United States, whether we are comfortable knowing this or not, we have drug and multi-drug resistant TB. There were over 1,000 cases of drug-resistant TB in the US last year, 10,000, that's 10%. And we had 6 cases of virtually untreatable, extremely drug-resistant TB for which we have no drugs. And right now, in the world, we have poor drugs. With the drugs that we have, there are 8 to 9 million new cases of drug-sensitive TB every year. It takes 6 months of treatment with 4 drugs, each of which have their own toxicities, some blindness, some liver toxicities, and there's about an 80% treatment success rate globally. For multi-drug resistant TB, there's now about 500 to 650,000 cases estimated by the World Health Organization. The definition is that they're resistant to the first 2 best TB drugs out there, isoniazid, and rifampin. You then have to choose from 6 of the just horrendously bad drugs. One is cycloserine, which the patients call psychoserine, gives them psychotic episodes. There's kanamycin, which causes irreversible loss of hearing. Another drug, irreversible loss of sight. And you have to take these drugs for up to 24 months in order to cure this disease. There's only about a 48% cure rate. An extremely drug-resistant TB, which is now here in the United States and is now detected in every country in the world. You'd need much more than 24 months to treat these patients, and there are no drugs. They are trying everything in the armamentarium that we have to find drugs, and Cliff has been a leader in finding drugs that can rescue these people from certain death. How do you feel? I feel kind of anxious now. The biggest problem that we have in TB is that this is a pathogen that can live inside cells and can live outside cells. So we have to have drugs that not only can get to the interstitial fluids in our tissues to kill those bugs that are outside, but we need drugs that can penetrate the macrophage where it lives and replicates and kill the bug there. So it's pretty tricky to get a drug that can go through a macrophage membrane, that can waddle through the macrophage cytoplasm, can get into the phagolysosome where the TB organism is, and can survive there long enough to get inside the TB organism and then kill it. So finding new drugs for TB is a challenge. Because we have another issue with TB, this has confounded the problem of finding drugs, and that is that TB used to be the #1 killer of US citizens prior to 1950. And those of you who are as old as I am all have a relative who died of TB. This organism was the impetus for pharmaceutical companies to get into the business of drug discovery and development, and many of the first drugs that came out were specifically directed to TB. Every time we introduced a drug, within 6 months the organism became resistant to that drug. So now we know that we have at least a minimum of 3 drugs that we have to treat people with to avoid resistance that occurs, to penetrate those appropriate tissues harboring the bacteria, and to kill both replicating bacteria and a trick that Mycobacteria does. It goes dormant, lies there unassumingly, waiting for his chance to start replicating again, and we don't even know where it is. So the goals of new TB drug development include identification of drugs with novel mechanisms of action. We need to pair 3 novel drugs together to increase both tissue distribution and kill various bacterial forms. We need to reduce the regimen toxicity, which is a problem in drug-sensitive TB, but which is a terrific problem in MDR and XDR TB. And hopefully at some point we will be able to shorten the treatment time. Now this isn't a regulatory goal, and it's not the basis for approval of any drug, but it's certainly a public health compliance goal. So we're going to go in, Cliff is going to take over here, but I thought I've never actually told Cliff why we started working together. I mean, he probably has his own stories as well, but I thought I would just reveal to you what happened to put Cliff and Carol together 15 years ago. So at that time, Tom Kint was the head of intramural research here at the NIH, and from my academic career, he was both a friend and a colleague. I had a problem, and he had a problem, and we agreed to try to help each other. My problem was nearly trivial. My problem was that I had started Sequela, and someone wanted to make a documentary film, a subject of which has long left my mind, and I needed an office that looked like an office. I was running the company out of my home, and I needed a bookcase filled not with children's books, but with medical text. And he offered, we used his bookcase, which was very beautiful, as the background for that. His problem was more concerning. he had a very, very bright young scientist who was working in Hamilton, Montana, and who he had to bring back here to the NIH campus. And this scientist wasn't exactly happy to be coming back here. He had a very nice life in Hamilton, Montana, although I don't know how one does that, but nonetheless. And so Tom asked if I would introduce myself to Cliff and see if I could generate a project when he got here that would keep him interested enough. Tom was very concerned that Cliff would get here and then would leave the NIH, and he thought that the government needed Cliff. So I went to a Gordon conference on TB, and I introduced myself to Cliff, and we talked, and this whole process worked because he's still here. So anyway, so that's the story of why we are together. Now I'm going to let Cliff tell you a little bit about his lab.
So that wasn't in the script, Carol.
I know. Sorry.
So we're going to take just a second to briefly introduce each of our respective organizations and where this project fits in. And then we're going to wind the clock back 15 years and literally walk you through the project that happened. And I think there's nobody in my laboratory, and there's a few of you out there now, who was actually present in the laboratory when this project started, or even when we basically stopped working on it for a long period of time. So what my group does these days, and we've done different iterations of this and have different emphasis over time, is we try to build a pipeline. There's always talk at NIH about bench to bedside stuff, right? Well, I'm a chemist, so I don't do benches, we do hoods, right? So we talk about hood to bedside stuff. So we've tried to recreate the entire process of doing drug discovery to the point that we can do it at NIH within the intramural research program. And so a lot of the lab will work on things like high throughput screening, systems biology, whole cell screens, trying to understand what are vulnerable points in metabolism and how do we deal with this organism. And then we'll move into animal model studies. That happens to be a very cute little monkey there, so we do some things in monkeys, some things in mice, and using tools that we can apply in the clinic, like imaging analysis, PET CT scans, things, biomarkers, and we try to pilot clinical trial designs. And then we actually do clinical trials, starting pretty much with phase 2, where we try to take those things we learn from the animal studies and translate them directly into people. And I'm not going to talk about anything in the last 2 phases of this. I just want to point out that a lot of my group is engaged in that sort of activity these days. But back in 1998 or so, we really focused much more on the early part of the pipeline because we didn't have very much to go later in the pipeline at that point. And I wanted to show you this because I think it's important for 2 reasons. One, because this is my current group, and I love to make people in the group angry by pointing out that they're arranged in order of importance by discipline. So the chemists are all in the front, the biologists, and then the sort of animal models people languish in the back. And then all of the clinical work that we do is offshore, mostly in South Korea and China. And this is a part of my clinical team, and other visitors, that's not that big, that actually put these things into humans and get information that feeds back all the way back to the beginning of the pipeline. So we develop better and better tools and better approaches for introducing new chemotherapies into the clinic. And I also just wanted to acknowledge up front the people who really did this work. And the principal driver of a lot of this particular project was a postdoctoral fellow who joined me after finishing up at Oxford, Richard Lee, who was a very talented chemist and now runs his own very successful group that still works in TB drug discovery at St. Jude's down in Tennessee. And many other people made contributions. I'm absolutely sure I left people off of this slide because literally this was 15 years ago that a lot of this was really starting and that people were jumping in and out of it. And I also included the people in tech transfer that made fundamental contributions to setting this up right at the beginning so that at the end we had a product to show for it and we didn't get lost along the way. And I'm equally certain that I've left people off that list, but those are the ones that I can remember that dealt with us on a day-to-day basis in establishing this project up front. Carol?
So I'd like to just introduce you to Sequela. Leo Link and I founded Sequela with 2 other founders, by the way, in 1997 as a way to focus energy of a for-profit biotechnology company on a global health issue, which was tuberculosis. We've gotten a lot different in the last 15 years for a variety of different reasons. And now we have actually compounds that work on all of these different infections. We do drug discovery in-house. We played off of Cliff. Cliff taught us how to do combinatorial chemistry and high-throughput screening, and we brought it in-house. And so now we have a compound library of about 250,000 molecules of various different classes. And I have 2 fabulous Russian women chemists who are just beyond belief good at what they do. So we're currently focused specifically on 3 infections: Mycobacterium tuberculosis, 1/3 of the world infected. Lots of people get it annually. And WHO reports lots of multi-drug resistant TB. Helicobacter pylori infections has even better statistics, 1 in 2. That's really awesome, isn't it? Causes gastritis and gastric cancer. There are 2 million cases, 2 million deaths every year, and there's a lot of antibiotic resistance. And we won't talk any about Clostridium difficile, but we do have an early stage program on that. What we will talk about today is the compound that we ended up with Cliff, and he'll tell you how we got here, whose indication is currently TB. It's now in a Russian registration trial for multi-drug resistant TB and a phase 2 trial for drug-sensitive TB. It's an awesome drug. This just shows you how many colonies come out of the standard of care TB drugs at the end of a 4-month treatment, and this is SQ109. Lots fewer bacteria, so it's a good drug. But I also wanted to mention that it's a multifunctional drug. It has a very limited number of bacteria that it can work on, but it does work on Helicobacter pylori. And we've done actually a phase 2 proof of principle trial looking at the FDA-approved biomarker for cure of Helicobacter infections of the stomach. And in fact, we can pull the biomarker down to undetectable levels. So we're very excited about having another indication as well for this drug because it's a really good drug, and so far with almost no safety issues.
Right, so let's wind back the clock. So the project started with Ethambutol. Now, Ethambutol is one of the 4 first-line drugs used in TB chemotherapy. It's probably widely considered to be the least effective of those, the 4 drugs, but it's included in that regimen. And so at least 2 million patients receive about 360 million doses of this drug every single year. And because it's the least active, we thought about long and hard about what if we could improve on Ethambutol. Ethambutol came from a program then at the Letterly Laboratories of American Cyanamid before they became American Home Products, et cetera, et cetera. When TB was still a big deal in this country, in a program in the 1950s, they identified in a whole cell screen this compound, N,N-diisopropylethylene diamine, as an inhibitor of tuberculosis growth. And through a very laborious hand process, they went through and made 2,000 analogs of that, which was a big program at Letterly Labs in those days. It'd be a big program these days in any indication area. And what they ended up with was Ethambutol. And what they knew in 1961 when they disclosed this to the world was that Ethambutol was really active against lethal lung infections in mice, with human TB, with oral administration, and it was active in vitro, and it looked safe, and it really blocked the development of resistance when you did co-therapy in mice. And so clinical trials being what they were in the 1960s, in mid-1961, they disclosed this. By the end of 1961, it was in clinical trials in humans. And so less than 6 months from discovery to actual first dose in humans, which is pretty incredible and pretty hard to replicate these days.
Impossible.
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 But it also comes with some risks, right? And I think this was a good example of the risk, because in that very first clinical study, which involved about 150 patients who received ethambutol and 150 who received placebo, 18 patients developed a serious ocular toxicity as a result of that. And you can see just one example of that kind of ocular toxicity, and we understand the mechanism for this these days, but back then they had no clue what the mechanism was. And this hemorrhage here was a serious dose-limiting toxicity for the drug. And so the next rounds of clinical trials used sequentially lower and lower doses of ethambutol. The first trial was stunning. The efficacy was great, but the toxicity was unacceptable. By the time they dropped the dose low enough to get around the toxicity, the efficacy was pretty questionable at that point, which is how we ended up with the weakest drug in that 4-drug regimen. And so in 1998, Carol and I asked the question, can we do better? Perhaps irrationally exhilarated by the fact that the lunar prospector had successfully crashed into a crater on the moon and discovered water on the moon, we thought we can surely do better than they could do in 1960 in making something a better ethambutol. And just to really I was really trying to recreate my frame of mind back in 1998. And we, like much of the world that did medicinal chemistry those days, were totally infatuated with combinatorial chemistry. And the only analogy I can draw to that I think had a similar impact on popular culture was Beanie Babies, right? Everybody had to have Beanie Babies, and the more Beanie Babies you had, the better. And it was like this with combinatorial chemistry. The more compounds you had, the better. And surely if we had enough Beanie Babies, the demand would get so large that we could retire on that, so we should invest everything we have in Beanie Babies. And every pharma company in the world went down this same path. Now, 10 years ago, we started to burn out on that, and these days nobody really banks on combinatorial chemistry, but that was the mindset at that point in time. We had to have bigger and bigger libraries. So what is combinatorial chemistry? Combinatorial chemistry is often called split and pool chemistry, and basically it's a process of building things on a solid support. And so say I have a green marble, and I take 3 different reactions with green marbles, and in one I react it with yellow, one with blue, and one with red. At the end of it, in each pot, I've got just one molecule, but it's green and red, or green and blue, or green and yellow. Now, if I pool those all together, I have a mixture of all 3 of those, and then I split them out again into 3 different pots and react them again with a yellow, a blue, or a red dot. Then in each pot, I end up with discrete mixtures of 3 different things that I can pool back together and split again and react with another single monomer. So you can, in a way, not only make these things combinatorial by split and pool synthesis, but you can also track them. And there's an infinite variety of different fancy ways of tracking what's on an individual bead. So in this way, it becomes possible to make libraries of millions of permutations if you're clever enough to work out the basic chemistry that goes into how you make those little links between the molecules and how you code or decode them. And again, we did a variety of different iterations of this. Some use radio frequency transmitters inside of an individual bead. Some use DNA barcodes. I mean, there's an unlimited number of ways of keeping track of what's on in any individual bead, but I'm not going to talk about that today. What I am going to talk about is how we reduce that to practice for trying to look for a better ethambutol. And this is solid phase chemistry, so that means there's a solid involved. So this little ball is the best representation a chemist can come up with for a piece of plastic, right? So we'll just leave it as a piece of plastic at this point. And it's got some fancy spinach on it. In this case, this is called a rink linker that just links the plastic to whatever you're going to put specifically on this position of the molecule. And then you can activate that. And then down here is ethambutol, just in case you don't remember the structure from the previous slide. Then I can take half of ethambutol and sort of patch it onto that linker so that it looks like this. So half of ethambutol. Then I can take a linker that looks like this and put it on the ethambutol half so that I've recreated this 2-carbon unit here. And then I can put on the other half of ethambutol. So I've got now the entire ethambutol molecule made on a specific bead. And then I can reduce that and cleave it off the bead. And I've just created the world's most expensive synthesis of ethambutol. Right? That's all I've done. I've just recreated a synthesis of ethambutol. But one where I can control what's in each of those positions because I have a solid supported synthesis and I can radio tag or tag in some way that bead that the molecule is actually made on. So it doesn't take a huge leap of imagination then to just erase the halves of ethambutol that I put in and put R as a generic R substitution and a generic linker and a generic R2 substitution. And then you start to realize you can create a library of things that look like this, all immobilized on a solid support, all in a way that you can do split and pool chemistry with. So the structures here aren't important. It's okay. It just looks like a plate of spaghetti anyways. But this just gives you an illustration of how fast and rapid this is and how quickly you can get to astronomical numbers that are almost uncontrollable. So if I just take 58 different R1s, and those are just different amines there, and I mix them up with themselves in the 2 different positions, and then the linker with 12 different positions, there are 40,000 unique combinations of molecules that can come out of that library of compounds that look like ethambutol. So we can create lots and lots of things that look like ethambutol. Letterly Labs made 2,000, right? So we can do orders of magnitude better than Letterly Labs did in surveying what the chemical space is of stuff that will inhibit that same target as ethambutol. But when you couple numbers like that with the need to do assays against the organism, you realize it becomes a little bit difficult to screen that number of compounds against the organism itself. And so this dovetailed very nicely with a project that was going on in the lab at the same time, where we were looking at the transcriptional responses. And this is like in the dark days of microarrays, right? We were doing literally spotted arrays, right? So this is the old school way of doing microarrays. The story is the same, but I mean, you did discover things in those days. You just had an incredible amount of noise in your signals. And basically what we did was we took all the known inhibitors of TB, looked at lots of microarray patterns, and we matched genes that went up or down, and we tried to make sense out of it. We looked for things that were common. So what we could tell is that we could lump things based on the big impacts on the cell based on their transcriptional fingerprints. And with ethambutol in particular, there was a unique set of genes that was very sensitive to inhibition of that particular step that we could then pull out from that analysis and create a useful screening system. And so what we did was we took the genes that were the most sensitive to treatment with ethambutol, fused them to luciferase, and then transformed that into TB. And now we have a strain that when you hit it with ethambutol produces light. And that's the kind of tool that you need to be able to do things in a high throughput format so that you can actually screen lots and lots of compounds against that particular kind of organism. And this just shows you a dose titration with different inhibitors of cell wall synthesis, and then the light production, and then the light production with ethambutol here. So you get a nice production of light when you hit it with ethambutol. We have a nice assay that could be reduced to 384 well plates, so we could screen lots and lots of compounds. We had a nice way of synthesizing lots and lots of compounds, so we did it. That's Richard Lee up in the corner, who went after this first. It's hard for us to recreate these days how many actual analogs we synthesized. So we say it was 100,000. That's, I think, what we ended up publishing, where we had enough analytical data to actually support it. But we made many more than that in different pilot libraries and different iterations of the library. Those are just the ones that were well enough characterized to actually go into this. And then we screened them using a variety of relatively, at that time, sophisticated robotics, which seem rather primitive now that I look at the pictures compared to what we use these days. But that was the robotics of being able to do it. And you have a few technicians who kind of load plates into these and continue to stack compounds, pair compounds with organisms, and look for light production. And so after screening 100,000 compounds now, 50 times the number that was screened at the Letterly Laboratories, what we came up with was 65 of them that were reliably as good as or better than ethambutol. And so the red line on this bar shows you where ethambutol's MIC was, and these are now not light production. These are actually resynthesized compounds where we measure physically an MIC. And the best of those had an MIC that was nanomolar, so orders of magnitude better than ethambutol. Now, of course, the best ones always got some other problems, right? So this is just the first assay, and then we go through a variety of different things where we selected for what the best one was that appeared safe and didn't kill the eukaryotic cells and had some pharmacology, et cetera, et cetera. And I'm not really going to talk about that in any great amount of detail. Instead, what I'll tell you is, this is what we ended up with, SQ109. There's many, many papers about this at this point, so I'm not going to go through a lot of depth in the pharmacology. Carol will give you a little bit more about the preclinical and IND-enabling pharmacology we had to do. But this was our candidate that came out of it, and what we considered to be the minimum pharmacophore, which was sort of this NN-diethyl amine substituent in the beginning. Now, one thing we did achieve in the process of doing this was getting rid of these secondary hydroxyl groups, and that's because, as I mentioned before, there is a lot of toxicity with the parent compound, and that's due to its ability to form a stable chelate with copper, which is conferred by those hydroxyl groups paired with the amines that were there. And so we wanted to get rid of that and try to destabilize any potential metal chelates to avoid that toxicity. And so you see we've gotten rid of the hydroxyl groups, and we put a big bulky substituent here, so we don't expect to see the ocular toxicity that we saw with ethambutol from the beginning. So we succeeded, right? Well, kinda. And the reason I say kinda is because we did actually end up creating something that looked like ethambutol and that worked with a similar mechanism, but we actually slipped off mechanism just a little bit. So instead of hitting the target for ethambutol, and this is a very complicated schematic of what the mycobacterial cell envelope looks like, right? It's a very complicated heteropolymer that consists of 3 different units: peptidoglycan, covalently linked to arabinogalactan, covalently linked to these mycolic acids. And ethambutol, we know, blocks production or blocks cross-linking of this arabinogalactan molecule. And what we found in the end, and again, this is all published work, so I'll show you just a tiny bit of it that's more contemporary in the lab. What we found in the end was that actually we pushed the target upstream a little bit, and we were blocking this transporter that pushes these mycolic acids from inside the cell to outside the cell where they're attached to the cell envelope. And the evidence for that is, again, too complicated, I think, for this, too much detail for this talk, but I do want to point out that we discovered this, we settled on the molecule in about 2001, 2002, but it wasn't until 2012 that we actually firmly established which step in the biosynthesis we were inhibiting and what the mechanism was for that inhibition. And we did that through a combination of both biochemical studies, where we identified these particular molecules that were accumulating inside the cell when we treated with SQ109, and genetic evidence, where we raised mutants that were resistant to the drug, and then we resequenced them and identified the targets, and then ultimately showed that that particular protein, which was this efflux transporter molecule, was actually the target, and we'd slipped off the ethambutol mechanism slightly. So that's a little bit of a rub, and it's important because when you look at the in vitro data for SQ109, I mean, so Carol will say this in a positive light. From my purist academic hat, this is an annoyance because we didn't exactly hit the target I wanted to hit. But from the positive spin on this that Carol prefers, it still has activity against ethambutol-resistant organisms, right? Because we've actually moved the target by a protein in the biosynthetic pathway. So in vitro, this was a good drug. It has an MIC that is less than micromolar against many different clinical isolates, multi-drug resistant, extensively drug-resistant disease, many different physiologies, so against intracellular and non-replicating bugs, it inhibits growth. As long as the cell has to be making cell envelope, it seems to kill them pretty effectively. In contrast, it's very inactive against HepG2 and a number of other cell lines, so it doesn't kill eukaryotic cells. It doesn't even kill other bacteria. It's highly specific for Mycobacterium, Mycobacteria, the genus of organisms. And it's very difficult for the organism to acquire resistance to this drug because it's relatively large, and the surface of that pore that we're blocking actually needs a very specific mutation to get resistance to it. So it's actually better in terms of resistance frequency than rifampicin. And there's some in vitro evidence for synergy between RIF and INH, indicating that this might really be something that could shorten therapy in TB patients. In vivo, we knew this in a sort of standardized way. This could protect mice from TB-induced weight loss. This is a rather high-throughput crude screen that we do. As a single agent, we knew it could reduce the CFUs in mouse lungs by 1 to 2 logs. The activity in vivo, that made it at least 10 times better than ethambutol, so we achieved at least that much in terms of potency. And when you combine it, and Carol just showed you some of this, and I'm going to show you the actual numbers for it in just a second, when you combine it in the sort of 4-drug standard of care regimen, in replacement of ethambutol, you definitely clear mice much quicker than they would be otherwise. And now, I'm going to be the first one to tell you mice don't mean anything when you talk about human disease, right? So I'll show mouse data with a shame look on my face because I don't really believe this tells you anything about what we should expect in people. You don't know what's going to happen in people until you do the experiment in people, which is being done now. But if you do what we hope to do in people, we took ethambutol out of that 4-drug regimen and we replaced it with SQ109. What the mice do is clear the bacteria much more quickly. Now, this is about 3 months of treatment. And if that translated into people, that would suggest that we had achieved something that would shorten the duration of therapy in people from 6 months, as it is presently, down to about 4 months. So that's what we knew from the in vivo mouse stuff. And frankly speaking, if we'd been only working in the intramural program, that's probably about where we would have run into the first huge roadblock, which is how do we get enough safety and tox and GMP and GLP and all the rest of that stuff to be able to go into human trials. Carol?
Yes, that's why it's nice to have a company that has a chief medical officer who knows exactly how to do all of this stuff, what you need to do to get a drug from a mouse to a human. And what I basically want to tell you is that we wouldn't have been able to do this without the help of a variety of institutes at the NIH. I mean, this truly was a NIAID initiated project, but it flowed all through other institutes at the NIH and other organizations both here in the United States and also in Europe. This is a very expensive process. The estimates for taking a drug all the way through from beginning to end is now upwards of $1.7 billion. I can tell you that it doesn't really, if your drug is good, it doesn't really take that much because they're amortizing everything that didn't work. But it does take hundreds of millions of dollars. And so it's difficult to accomplish all the resources that you need to accomplish to move from point A to point B to point C in a systematic way in the clinic because the cost is so high. So there was a program called the Inter-Institute Program between the National Cancer Institute, which at that time had a number of contractors that were doing work for the Cancer Institute itself and for people who were working with the Cancer Institute in academic institutions, and NIAID. So it was a joint program, and it enabled NIAID to select one drug that the Inter-Institute Program would work with at the Cancer Institute with their contractors to help move it forward. And we won the contest one year. We were very excited. And so we ended up working with the Inter-Institute Program and a variety of awesome people at the NCI itself and also in their contracting groups. And these are just some of the studies that had to go on before we moved this drug into first-in-man studies in human. I guess the one that's not on here that we did that was the most expensive is one that Sequela actually did. This drug likes to be inside of cells. It likes to be inside of exactly the cells where TB hides. And when it gets in there, the cells wrap it up in a lipid vesicle that's called phospholipidosis. And so there are about 35, 40, 50 compounds out there that cause the same kind of thing. And for a while, we thought phospholipidosis was a toxic effect. Now we know it's a protective effect. And what it actually does is it causes the drug to get into cells and then to be released out into the circulation slowly over time. So, but at the time that we submitted our IND, we had 90-day rats, 90-day dogs. Posaconazole from a big pharma company had just gone through the FDA, and they actually demonstrated that there is no toxic effect of phospholipidosis for posaconazole. And in fairness to the pharma company, we were then, and they demonstrated this in monkeys. We were then asked, because they didn't want to ask one company to do this and not ask another, to do a monkey study, which ended up costing $3 million. And delayed the onset of the human clinical trials by about a year. So we also found no toxic effects of phospholipidosis. And the FDA, and we convened a panel, including the posaconazole people, and the FDA does now, they're interested in in watching it, but they're not rejecting drugs on the basis of phospholipidosis. And with that $3 million, I think helped do that. It's very annoying, I must say. So, so we did start first-in-man studies, and the first set of studies Sequela did. This was in 2006, and we were completed this study in close to the end of middle of 2007. And as you may recall, in 2007, the economy collapsed, and so we talked to the NIH, and they agreed that we could conserve our money so we could stay alive as others, and NIH actually ended up doing the rest of the phase 1 studies. We had no toxicity in patients up to 300 milligrams. It was really lovely to see these 14-day studies. We then went on to do a phase 2A safety study in TB patients, and this is funded by actually the European Union through a program called EDCTP. And the African sites where this was done was called Panacea. So we now have more people involved in development of this drug. This is the data. The data would tell you that all these are SQ109 by itself. This is SQ109 in the presence of rifampin, and that's rifampin, SQ109 and SQ109 plus rifampin. So in fact, in the first 14 days of patient treatment, there's not a lot of activity of SQ109 for TB. But we had already done the mouse study, and we already knew that. So we consider the phase 2A a safety study in human patients. And in fact, we don't see any activity of SQ109 for the first 3 weeks. Starting at 4 weeks, we start seeing significant activity, and then it goes on from there. So we are now, well, again, safe. The only thing is some nausea. It doesn't taste very good. And so, but if you feed people while you give them the drug, they're fine. We know a lot about the drug now in TB patients. It's equally safe in TB patients and healthy individuals. So now we have some ongoing clinical trials. We licensed the rights to sell this drug in Russia and the affiliated states with Russia to Infectex, a company that was actually created around SQ109 in Russia. And they have an ongoing trial for registration of this drug for marketing and sales there. And of course, the EDCTP funded phase 2B study looking at longer duration of treatment of drug-sensitive TB is done with Panacea and funding from the EDCTP. We've got lots of partners, mostly in Southern Africa and in Tanzania, doing these studies. And there's a lot to do left before we can do a definitive trial for registration of this drug. We've got them all planned. We've got them costed. We're just out raising money at the moment. This is the timeline for getting SQ109 to the FDA. I'm raising $38 million at the moment. If anyone has any millions they don't know what to do with, see me after. But that's the timeline. We're hoping by 2017 this drug will have gone through its paces and will actually be out in Russia in marketing and sales. I think you're next.
Okay, I think that was Carol's TB flaring up again.
Yeah, it is. Don't worry. I have a drug that can cure you.
And if anybody would like to taste the SQ109,
yeah, that's right.
So SQ109, I mean, it's a long process. This is not atypical for TB drugs, right? It takes years, it takes decades often to come up with how a particular drug fits into that regimen. And I think the drug's got a lot of good things going for it. It might still bomb in phase 2B, We can't, there's no guarantees in this game. We take big, expensive bets, and we try to make the best guesses we can and do the best animal studies we can to assess efficacy before we get to it. But that's these things can fail for any number of reasons still. But nonetheless, we never would have gotten to the point of having the ability to test this in humans and to put this idea together without the team of people that really gathered around the initial idea and to translate this into a product. And so, once again, I want to take the opportunity to thank that whole team and to thank Carol and I guess Tom Kint for making it possible to actually turn something. I mean, it's quite gratifying, I have to say, as more of a bench basic scientist guy, to be able to put your hand on a pill and say, I made this. And we actually did do that, and we made it and Carol made it possible, and Sequela made it possible. And behind all of that, OTT and Phil Chen made this possible. And I think, again, I think both I and Carol want to thank OTT and Phil and Mark and the whole gang for sort of putting this together right from the beginning, so we got the chance to take this big gamble and put this into people. And we hope that in the future, TB patients will flood you with letters to thank you for that treatment. So I think we'll end there and say thanks for your attention, and thanks for the honor of doing the Phil Chen lecture.
I think we could have a couple of questions. It's a little late, but let me start. So you gave us some indication that the target was not the same as Ethambutol. Can you give us a little more about what the target is and how it is toxic to TB?
Sure. It's just literally 2 steps removed from the target for Ethambutol. So we do think it's related in the way it works, but these are these macromolecular complexes that make the cell envelope sort of talk to the cytoplasm. And it's at the point where the precursor is pulled in from the cytoplasm and inverted before it's attached to the growing cell envelope. That's where the drug actually blocks that efflux system.
It's a cell wall active drug.
It is, yeah. I love that.
Yeah,
I know.
But I should say, while yeah.
It's right up your alley.
Right. While Cliff is, I just want to answer also, I want to make a statement. While Cliff, it was disturbed that we started out to make a second generation Ethambutol and we ended up with a not Ethambutol. That actually created the opportunity for a patent, because Ethambutol was already patented and was off patent. And so it would have been an obvious thing if we had created a new drug. So while I am delighted that we have a new mechanism of action, and it's good because we can now not worry about Ethambutol resistance in multi-drug resistant TB when we do our treatments. That's all I wanted to say.
Thank you for sharing this wonderful story. I have a question. We're seeing an emergence of a new requirement that the preclinical work will have to be authenticated by an independent entity before going into patients. I'd love to hear your comments to your reaction to how that would impact the pace of this translational process, in your opinion.
When you're asking about, when you're talking about preclinical work done in an academic center or done here, or?
I don't think it matters where it's done. If you're doing, there's a question of the validity of the preclinical work. People have sort of done surveys broadly across products that have been evaluated for clinical work, and there's been some question of the authenticity of the product that was actually brought to translational into the patients. And so there's discussion about authenticating the preclinical product to make sure all the work is indeed what it's thought to be.
Well, so if by preclinical you mean non-clinical, the discovery part of it, I can understand that completely. On the preclinical side, it's all done GLP and it's all checked, and the FDA is very rigorous about looking at it as well. So you don't get into humans unless there
I'm talking
are
about the
validated
discovery
assays.
side.
On the discovery side, quite honestly, if I had any advice for universities, I would ask them to begin teaching people how to do research under GLP light, good laboratory practices light, because that's the biggest problem. If you don't have someone checking your lab book, if you don't have validated assays, by the time it comes to me, it takes me several years to re-engineer it and to figure out whether you were right or not. So it's a process. There is not, I've never seen a technology come out of an academic center right into a small company and be ready to go into humans. There's always lots of things we have to do to validate the process. So the more you can use good laboratory practice, and it's a painful thing, and the more you can teach your people to do that, the easier it will be to translate it, the faster we'll get to humans.
So let me take a stab at that too, because I think Carol has one perspective on it, and I have a different perspective. I mean, you can look at that,
Really?
that timeline of antibiotic discovery, right, that we show at the beginning, and we can call that an innovation gap if we want, because there are no new antibiotics that were introduced into clinical practice between 1970 and the year 2000, right? No new class. We had lots of knockoffs of things. What really changed was the culture at FDA. Selman Waxman discovered streptomycin and within 4 months, the first patient at the Mayo Clinic had received a dose of streptomycin. But we've become so risk averse, and FDA is so harsh on preclinical IND-enabling tox and all the logic around getting into a phase 1 that we've killed antibiotic discovery. There are almost no major pharma programs currently in anti-infectives. The reason? It's too hard. It's too hard to get something that it's too expensive to take that shot, to get into humans, to fail in humans, and we've erected all these barriers. So I think I take the opposite view. We shouldn't learn how to do GLP. We should learn how to reinvent the clinical trials so we can get into humans faster, and reduce the regulation in the clinical trials so we can get into humans faster.
And I think risk is the biggest issue. I mean, we have, the FDA pendulum swings back and forth. We're sort of in the middle now. It's a lot easier. Our discussions with them, they're a lot more open for discussions. I have no problem with the FDA right now in what they're asking us to do. There was a point at which they got very cranky about any safety issues. And what they're now looking at is risk and benefit. And I think that's really important. There is nothing that you put in your mouths that doesn't have risk, including water. But we've gotten to the point in our communities where we don't want risk. And that's why there are parents all over the place not vaccinating their children, because 1 out of every 500,000 babies has a problem with vaccination. It's, there is risk with everything that we do. And I don't want to go back to 4 months before we get it into humans. I already know that that's not good. But from the perspective of actually taking the risk, actually having the risk once you put the product out there. But I don't want to have a 15, 20-year development cycle either. There has to be a balance.
I want to ask about the regulatory challenges of developing a drug that's going to be used in a multi-drug combination therapy. So I noticed that in your phase 1 that you tested just the SQ109. Were there discussions with FDA? Did they want to try it in combination with the other drugs that you might be using in phase 1? And then along with that, in your clinical trials, in this case, you can kind of slip it in for EMB because it's a similar kind of pathway. But, so how do you work it in with the combination? Do you add it on, or do you substitute and so forth?
Sometimes you add it on. Sometimes you replace something. The first-in-man studies, you just have to demonstrate that your drug is safe, that it's not going to kill people, and it's not going to cause them irreparable damage. It's okay if it causes them a little damage and they get better. So in the first-in-man studies, you always do just the drug alone. The phase 2A study, which I think of as a TB patient safety study, we did add it into rifampin because we actually were interested to see whether in a short period of time we could make rifampin look better as it does in the mice. 14 days is too short, as actually we knew in the mice. And now we're doing drug-drug interaction studies in healthy humans and TB patients, because TB patients are sick, and they're going to respond differently. So it's a step process, and at each step, you try to reduce the risk for the people that you're using as your guinea pigs. They are not guinea pigs. They're human beings. And so it's a process of trying to figure out risk-benefit for each of them and moving it along carefully so that you always know safety. Safety is the principal issue for FDA. Efficacy is important, but safe and efficacious. Safe and efficacious.
Hi, I wanted, that was a great lecture. Thank you very much. I
Thank
wanted
you.
to ask a question about whether there is any role for considerations of price point when you're developing drugs for scourges that are very widespread in developing countries, have to be taken for a long time and so forth. I know Cliff mentioned that he made the most expensive Ethambutol with his, original Combichem approach, but, for the final product, how did that come out? And you need the drug first, I understand, but
No,
is that a consideration at all?
Yeah, well, of course. Antibiotics are not biologics, and we can't sell them at $300,000 per course. It just doesn't work. People won't pay that. So price point is important. We've now, that was a Combichem program. We've now done solution-based chemistry and manufactured all of our batches that we need for the FDA, and the price will go down even further. It's way down now, but it'll go down even further once we scale up for commercial production, if we get to do that. And are there any venture capital firms in here? Are there any funders of high net worth investors that like to fund people here? Okay. All right. So from my perspective, it depends. The price of my drug depends on who I'm talking to. If I'm raising money, I'm going to tell them that the market is X because something is sold at that price. So linezolid is sold at $100 a pill. I will say it's $100 a pill. I will know in my heart of hearts that we probably won't be selling it for $100 a pill, but I might. There is that chance. And for a funder to know that the market opportunity is big means that they'll give me the money to get it to the point where I'll know what the price will be. So thank you for not having any venture capital people here. And don't publicize this to any venture capital people.
You're being televised. And
I know, oh, no.
Let's take one final question, and then we'll have a reception in the back. You can meet with Phil and talk to the speakers.
So thank you very much for the lecture. My question was more to do with the slide before this, showing that as far as technology transfer and drug development is concerned, you need a lot of partnerships. And I noticed that you work in Tanzania. What would you say you've had a very successful partnership in being able to bring all these different people together? What would you recommend or suggest or say are the most important things to consider when you're trying to do something like this in a place like Tanzania?
I think the most important thing for any set of relationships is to understand the role that each group plays and to have respect for the work that they do. If you do that, including the patients, by the way, if you can understand what the agenda is of every group and it's open and transparent and you talk about it, the things that I need to do for my drug for the FDA are going to be quite different than a clinician in Tanzania wants to do for a patient who is desperate. So we have to talk, and we have to communicate. We have to understand the agendas of everybody, and then we have to try to make a plan that works for as many people as we can. And we've had great success with the Panacea group, and the principal investigator for our particular study is Michael Holscher, who has a whole site in Tanzania.
All right. So first of all, let me thank the speakers for a wonderful coordinated presentation. Thank Phil and OTT for making this all possible. And there is a reception in the rear. You can talk to the speakers. You can talk to Phil. and you can share your thoughts about drug development. Thank you.